everybody, and today we are going to be talking about mitosis and meiosis. Let's get started with some definitions. Mitosis is the process of cell division in which a single parent cell divides into two daughter cells. If we were to draw it out, it'd look like this, with our parent cell and our two daughter cells. Moving on to meiosis. Meiosis functions to produce gametes by making daughter cells with exactly half as many chromosomes as the starting cells. Meiosis in humans is a division process that begins with a diploid cell containing two sets of chromosomes and ends with haploid cells containing single sets of chromosomes. So if we wanted to make this into a diagram, we'd have our parent cell here and our daughter cells, and then these haploid cells. You can see that these are the chromosomes. Now there are phases of mitosis and meiosis, which are prophase, metaphase, anaphase, telophase, and cytokinesis. Mitosis has 5 stages, while meiosis has 10 stages, or 4 and 8 if we don't count cytokinesis. Starting with prophase in mitosis. Prophase is when the chromosomes undergo the condensing process, which is a process that will continue to the metaphase. Metaphase is when the chromosomes line up to the middle of the cell, and the spindle fibers attach to each chromosome's centromere. During this phase, chromosomes temporarily stop their movement, at a point that ensures that only correctly assembled chromosomes can move on to anaphase. In anaphase, sister chromatids separate and are pulled to the opposite poles of each cell by the spindle fibers, which leads to telophase. Telophase is when the chromosomes reach opposite poles and a nuclear membrane forms around each set of chromosomes. The cell then splits into two through cytokinesis, creating identical daughter cells. A similar pattern of phases happens in meiosis 1, but after that there's an additional step known as meiosis 2. This contains prophase 2, metaphase 2, anaphase 2, telophase 2, and cytokinesis 2. The main difference between phase 1 and phase 2 is that the chromosomes are attached as homologous pairs on phase 1, but in phase 2, single chromosomes are attached to the equator, and through that, the haploid cells are formed. Let's go over some similarities and differences between mitosis and meiosis. Some similarities include that they both produce new cells. Mitosis produces 2 diploid daughter cells, and meiosis produces 4 haploid daughter cells. They also both start with a single parent cell. The difference would be that mitosis has 4 stages, 5 if you count cytokinesis. Mitosis happens in somatic cells. It produces 2 diploid daughter cells, but the genetic variation does not change. Meanwhile, meiosis has 8 stages, and 10 if you count cytokinesis. It happens in germ cells. It also produces 4 haploid daughter cells, and genetic variation is increased. Let's go to an example. Which of the following are the similarities between mitosis and meiosis? A. They both produce diploid daughter cells. B. They both produce genetically identical cells. C. Sister chromatids segregate to opposite poles of the cell during both processes. Or D. Crossing over occurs between chromatids during both processes. Think about your answer now. The correct answer will be revealed shortly. The answer would be C. Sister chromatids segregate to opposite poles of the cell during both mitosis and meiosis. A is incorrect because mitosis produces diploid daughter cells, while meiosis produces haploid daughter cells. B is incorrect because mitosis produces genetically identical cells, but meiosis produces genetically diverse cells due to crossing over an independent assortment of the homologous chromosome. D is incorrect because crossing over occurs between homologous chromosomes during meiosis, but not during mitosis. And that is it for our video on mitosis and meiosis. Thank you so much for watching. Make sure to like and subscribe.